Today in class, you were assigned page 13 from the packet. This is page 13 in your packet. You have various cells. You have to identify what phase of the cell cycle they're in. You also have to identify various structures within those cells. I have added a word bank for you so that you can easily identify those structures. Notice that centrosome or centriole is going to be used twice in this handout. Also in class, we completed the cell cycle lab. So this website is posted in the Google Classroom. You also got the handout that goes with this. The directions are on the handout. Once you watch the video and read the information, you click on the microscope and you're going to view 6 different slides with cells on them. This is normal lung. In order to switch between slides, you have to click and hold on the tissue slides, and then you just move between them, picking whichever tissue sample that you're going to look at. Once you have the tissue sample in front of you, you have to look at the cells and decide what phase you think they're in. So this cell here, I see chromosomes lined up along the center, and if I look above at these pictures, metaphase looks like the chromosomes are lined up along the center. So you have to click on that image and then take the word metaphase and drag it down into the little box. This cell here to me looks like it's in telophase. I see that dividing of the cytoplasm there. So I drag the label down to the box. This one here looks more like anaphase. I don't really see that indentation that I see in telophase. So I'm, again, clicking the label, dragging down to the box. Down here, I see that distinct indentation. I'm starting to reform that nucleus inside of there. So I would say that's telophase. And down here, this one looks like it's in prophase. I can clearly see those distinct chromosomes, and they're not lined up along the center. So once you've put labels in each of the little boxes, you hit FEC. If it's right, it gives you that noise. If it's wrong, it kind of buzzes at you, and you can switch out for the correct labels. Now that you've checked it, you need to add up how many cells you have in each phase. So how many cells are in interphase, how many cells are in anaphase, how many are in metaphase, etc. Once that you've counted however many cells that you have, you're going to fill that in on your lab handout. So let's just say that we were counting for the normal lung, and we counted 16 cells in interphase of the normal lung. And let's say that we counted 2 cells in prophase. Let's say 0 cells were in metaphase. 1 was in anaphase. And 1 was in telophase. And those are not the correct numbers. I'm just using those numbers as an example. So once I've completed one slide, you can either go through and complete the remaining slides, or you can come here and perform your calculations. You have to do all of it though, so it does not matter what order you do it in. For the calculations, I need to calculate the percentage of cells that are sitting in mitosis and the percentage of cells sitting in interphase. Mitosis consists of prophase, metaphase, anaphase, and telophase. So I need to add up all of those phases, prophase, metaphase, anaphase, and telophase, and then divide them by the total number of cells that were in the slide to begin with, that were in the sample, times 100, because it is a percent. So if I add up prophase, metaphase, anaphase, and telophase, I have 4 cells. Again, I'm dividing that by the total. So the total here, 16 and 2 plus 2 more, gives me a total of 20 cells. So for this row, I said that I had 4 that were in prophase, metaphase, anaphase, telophase, divided by a total of 20 times 100, which gives me 20%. So I would come back to my handout and I would label 20% on my handout. I'll do the same calculation for the cells that are in interphase. For the interphase cells, I don't have to add any other phases to it. Interphase is just itself. So how many cells are sitting in interphase divided by the total number times 100. So in this case, we had 16 cells in interphase divided by a total of 20 cells times 100. And that's going to give me a total percentage of 80%. And I'll fill that in on the chart. And you should notice that your two percentages will add up to 100. If they don't, something must be wrong with either the ones that you counted or maybe your addition or even how you set up your equation. Once you fill in all of your percentages, you'll come down below. You have 3 questions that you're answering. These 3 questions, you're really basing that off of the data that you collected. So how many cells did you have sitting in interphase? How many cells did you have sitting in the various stages of mitosis? What percentage of your cells are in mitosis? What percentage of your cells are still in interphase? Again, you're going to do that comparison when you're looking at number 2, but now we're kind of just focusing on the cancer cells. So only look at the rows that say cancer. So the cancerous lung, the cancerous ovary, the cancerous stomach. And then finally, again, you're looking at all the cells, and it talks about the mitotic index. Mitotic index is referencing that percentage of cells that are sitting in mitosis, and you're comparing the cancerous cells to the normal cells. So make sure you do all of your calculations before you answer these analysis questions.